Hello everyone. In this video, I'll show you how to install FastQC, create and activate our conda environment, verify that FastQC is working, and finally run FastQC on a sample FastQ.gz file. On this page, I'm explaining what conda is and how it works internally, especially for computational biology and bioinformatics workflows. What is conda? So first question, what is conda? Conda is both a package manager and an environment manager. It solves two major problems in computational science. First, software installation. Second, dependency conflicts. In research, different projects often need different software versions. Without conda, managing these versions becomes very difficult. Why conda is needed? For example, Project A may need Python 3.7 with NumPy 1.18. Project B may need Python 3.11 with NumPy 1.26. Without conda, maintaining both setups on the same machine is almost impossible. But with conda, each project lives in its own environment, so nothing breaks. How conda works internally? Now let's understand how conda works internally. You request a package. For example, you run a command to install FastQC or Python. Conda checks your system. It checks the operating system, whether it is Linux or Mac. It checks system architecture and Python version. Dependency resolution. Conda builds a dependency graph. This means it identifies all libraries required for that package. It resolves conflicts. If two packages need different versions of the same library, conda decides a compatible combination. It installs compatible libraries. This process is called as a T-solver. Real-world usage. This is why conda is widely used in genomics, machine learning, HPC environments. I am showing you a complete conda workflow that we commonly use in bioinformatics projects. First step is installing Miniconda on Linux. We download it using wget, run the installer, and then confirm the installation by checking the conda version. Next, we configure the channels. This is very important. We add conda-forge and bioconda, and set the channel priority to strict so that bioinformatics packages install smoothly without dependency issues. After that, we create a dedicated RNA-seq environment using Python 3.10 and activate it. I always recommend creating separate environments for each project. It keeps your system clean and your analysis reproducible. Then we install essential bioinformatics tools like FastQC, HISAT2, Samtools, and StringTie directly inside this environment. And finally, we verify the installation by running help commands for each tool, just to make sure everything is working correctly. Alright, now that conda is installed and our environment is created, this slide is about how to manage environments properly, and how to make your work reproducible. First, if you want to see all environments on your system, you can run conda env list. This shows you your base environment and any project environments you've created. To start working inside a specific environment, we activate it. For example, conda activate RNA-seq env. And when you're done, you can simply exit the environment using conda deactivate. Now, sometimes you want to check what tools are installed inside your environment. For that, use conda list. This will show all packages and their versions. If you ever want to delete an environment completely, like if you don't need it anymore, you can remove it using conda remove nrna-seq env, all y. Now the most important part here is reproducibility. Recreate the exact same setup later, you can export it to a YAML file. Conda env export greater than rna-seq env.yml. Now that we've understood the theory behind conda, let's move to the practical part and actually run everything step by step. First, I'm checking whether conda is installed correctly on my system. I run the command to check the conda version, and you can see it confirms that conda is available and ready to use. Next, I create a new environment specifically for RNA-seq work. I'm naming it RNA_seq_work and installing Python 3.10 inside it. This ensures that all tools we install will remain isolated from the base system. Once the environment is created, I activate it. Now you can see the environment name appears in the terminal, which means all installations will happen inside this workspace. Now I install commonly used tools like FastQC and HISA-T2 directly using Conda. These tools are pulled from Bioconda and conda-forge repositories, which are widely used in bioinformatics. This is the easiest way to install software without manually resolving dependencies. After installation, I check whether FastQC is working by running the help command. You can see the FastQC manual appears, confirming successful installation. I do the same for HISA-T2 to confirm it's installed correctly. Next, I configure Conda channels by adding conda-forge and bioconda and setting the channel priority to strict. This ensures future installations use the correct repositories. Now comes the actual analysis step. I navigate to the folder containing the FastQ file. Then I run FastQC on the sequencing file. You can see the program starts processing and shows progress as it analyzes the data quality. When we open the HTML report, we can see multiple quality metrics. This includes basic statistics, per base quality scores, GC content, and adapter content. These plots help us decide whether trimming or preprocessing is required before alignment. After finishing the analysis and installation steps, the next important practice is to deactivate the environment. This helps us return to the base system and prevents accidental installations in the wrong environment. So here, I run the command conda deactivate. After deactivating the environment, I am now showing how to remove an environment completely if it's no longer needed. This is useful when a project is finished or when you want to free up space. To delete the environment, we use the command conda remove nrna_suck_work, all y. So this is how we move from theory to real bioinformatics analysis, using conda to create environments, install tools, and perform sequencing quality checks using FastQC. This workflow is the foundation of almost every RNA-seq, ATAC-seq, and genomics pipeline. Before we end, let's quickly summarize what we learned in this video. We started with understanding conda and why it is important in bioinformatics. Then we installed Miniconda, configured channels, created a new environment, and installed tools like FastQC and HISA-T2. After that, we ran FastQC on sequencing data and interpreted the quality report. This workflow is the foundation for most RNA-seq and next-generation sequencing analyses. In upcoming videos, we'll move forward into more practical steps, including read trimming, alignment, RNA-seq pipelines, and advanced bioinformatics workflows using Conda and HPC. Thank you for watching this tutorial and being part of this learning journey. If you found this video helpful, please like the video, subscribe to the channel, and share it with others who are starting in bioinformatics.